Welcome to Principles of Ecology. Today, we are diving into the fascinating world of physical factors, specifically focusing on temperature and light. As a zoology student, understanding these non-living, abiotic components is absolutely crucial. They are the invisible forces that dictate where organisms can survive, how they behave, and how entire ecosystems function. By the end of this session, you will see the natural world through a completely new lens, understanding exactly how the physical environment shapes the biological one. Let us begin our journey. Now that we know our focus, let us start with our first physical factor, temperature. Imagine stepping out of a heavily air-conditioned room into the blazing summer heat. Your body reacts instantly. In ecology, temperature acts as a master dial for all biological activity. It is formally defined as a measure of the average kinetic energy of molecules in the environment. This energy directly influences the metabolic rates of all living organisms. Because of this, temperature is a primary abiotic factor that determines the geographical distribution and physiological performance of species across the globe. It dictates whether a seed sprouts, a bear hibernates, or a coral reef thrives. Building on that idea of metabolic rates, we must ask, why does temperature matter so much at a microscopic level? The answer lies in enzymes. All biological processes are driven by chemical reactions, which are catalyzed by these protein molecules. Enzymes are incredibly sensitive to temperature. If it is too cold, they work too slowly to sustain life. If it is too hot, they denature, losing their shape and stopping entirely. Because of this, organisms have specific temperature ranges, consisting of a minimum, an optimum, and a maximum. When environmental temperatures fall outside this optimal range, organisms experience severe physiological stress, leading to reduced growth or even death. Building on the importance of optimal temperature ranges, we must consider the broader ecological implications. Why does understanding these thermal limits matter so much today? The answer is global climate change. As environmental temperatures shift, ecologists use this knowledge to predict how species will respond. Some organisms will be forced to migrate to cooler areas, shifting entire ecosystem boundaries. Others may attempt to adapt to the new conditions. Unfortunately, species that cannot migrate or adapt fast enough face the tragic reality of extinction. This makes temperature not just a biological variable, but a critical factor in modern conservation efforts. To visualize these temperature ranges we just discussed, let us look at the thermal performance curve. On the horizontal axis, we have the environmental temperature in Celsius. On the vertical axis, we measure biological performance or growth rate. Notice the distinct bell-shaped curve. It starts at the minimum temperature, peaks beautifully at the optimum temperature where the organism thrives best, and drops sharply toward the maximum temperature. The green shaded area under the curve represents the zone where the organism can successfully survive and reproduce. Anything outside these dashed lines represents extreme stress or death. This graph perfectly illustrates the physical limits placed on life. Understanding these thermal limits naturally leads us to wonder how we can measure this sensitivity mathematically. This is where the Q10 temperature coefficient comes in. Conceptually, this formula calculates the factor by which the rate of a biological process increases for every 10 degrees Celsius rise in temperature. Let us break down the symbols. Q10 is the coefficient itself. R1 is the metabolic rate at the lower temperature, T1. R2 is the metabolic rate at the higher temperature, T2. If Q10 equals exactly 2, it means the organism's metabolic rate doubles for every 10-degree increase. The exponent mathematically adjusts the ratio of the rates to standardize it to a perfect 10-degree window, no matter what the actual temperature difference is. Before we move on, let us see this formula in action with a concrete example. A biologist is studying a cold-blooded frog. We are given an initial temperature of 15 degrees Celsius with a respiration rate of 4 units per minute. The temperature is raised to 25 degrees, and the rate increases to 8 units per minute. We need to calculate Q10. First, we substitute our values into the formula. The ratio of the rates is 8 divided by 4. The temperature difference in the exponent is 25 minus 15, which is 10. The exponent simplifies to 10 over 10, or 1. 8 divided by 4 is 2, so Q10 equals 2. Physically, this means the frog's respiration rate exactly doubles for every 10-degree increase, showing its direct dependence on environmental temperature. Now that we have thoroughly explored temperature, let us shift our focus to our second major physical factor, light. Have you ever noticed how a potted plant bends toward a window? Light is the ultimate source of energy for almost all life on Earth. Formally, it is the visible portion of the electromagnetic spectrum that reaches the Earth's surface. In ecology, light is the primary driver of photosynthesis in autotrophs, allowing them to convert solar energy into usable chemical energy. Without this constant influx of solar energy, primary producers could not create glucose, and the entire ecological food web would starve. It is the absolute base of the ecological energy pyramid. Beyond just providing energy for photosynthesis, light plays several other critical roles in an ecosystem. It serves as a crucial environmental cue for behavioral and developmental processes. The duration of light, known as the photoperiod, acts as a biological clock. It tells birds exactly when to migrate south for the winter and signals trees to drop their leaves in the fall. Furthermore, in aquatic ecosystems, light penetration is the defining factor for life. It determines the photic zone, which is the only layer of water where photosynthetic life can exist. The availability of light changes drastically depending on the physical structure of the habitat, structuring the entire underwater community. Since light penetration is so critical for aquatic life, we need a way to calculate how light fades as it travels through a medium like water. This brings us to the Beer-Lambert law applied to ecology. Conceptually, this formula states that light intensity decreases exponentially the deeper you go. Let us look at the variables. Iz represents the light intensity at a specific depth z, measured in meters. I0 is the initial light intensity at the surface. The constant E is the base of the natural logarithm. Finally, K is the light extinction coefficient, which measures how quickly the water absorbs or scatters light. If the water becomes very murky, the value of K increases, causing the available light to drop off much faster, severely limiting the photic zone. Now that we have our formula, let us apply it to a real-world scenario. A marine biologist is studying a clear freshwater lake. We are given a surface sunlight intensity of 1,000 units. The extinction coefficient for this clear water is 0.1 per meter. We need to find the light intensity at a depth of 10 meters to see if algae can survive there. We plug our values into the Beer-Lambert law. The exponent becomes negative 0.1 multiplied by 10, which simplifies neatly to negative 1. 1,000 multiplied by e to the power of negative 1 gives us approximately 368. Physically, this means only about 36.8% of the surface light reaches this depth, which is likely enough to support shade-tolerant algae. To truly grasp the result we just calculated, let us look at a visual representation of light attenuation. In this diagram, the horizontal axis represents light intensity, while the vertical axis represents depth, pointing downwards into the water. Notice the curve. It is not a straight line. It is an exponential decay curve. At the surface, light is at its maximum. As we move deeper, the intensity drops off rapidly. The diagram clearly marks the photic zone at the top, where enough light exists for photosynthesis, and the aphotic zone below, where it is too dark. This visual perfectly maps the physical limits placed on photosynthetic organisms by their environment. We have covered a lot of ground today, so let us summarize our key takeaways. First, we learned that temperature acts as the master dial for ecosystems, controlling enzyme efficiency and dictating where species can survive. We used the Q10 coefficient to mathematically measure how sensitive metabolic rates are to these temperature changes. Second, we explored light as the energetic foundation of life and a vital signaling mechanism for biological rhythms. Finally, we applied the Beer-Lambert law to understand how light intensity drops exponentially in environments like deep lakes. Together, these physical factors beautifully shape the structure and pace of life on Earth. Excellent work today.